complex movement in a context of general movement. Okay, so that's what we talked about last time. And okay, sorry. Okay, so today we're going to talk about two topics. One is the upper motor neuron brainstem projections. Okay, remember those are the sort of second half of the big system overlying the lower motor neurons. And then we're going to talk about one of the two circuits that influences those upper motor neuron circuits, and that's going to be the basal ganglia circuits. Okay, and like with the retina and the horizontal cells, I hope you had your coffee, because we're going to talk a lot about sine inversions and cool networks. So it'll be fun. And today we're going to actually get to something that I think motivates a lot of people, which is different aspects of neurodegeneration. We're going to talk a little bit about Parkinson's and Huntington's disease, which is related to degeneration in the basal ganglia circuits. All right. So this part of the circuit, the upper motor neurons of the cerebral cortex. Those were the M1 neurons. We talked about how they had a direct line down to the lower motor neurons. And now we're going to talk about the second class of upper motor neurons, and these are the upper motor neurons of the brainstem. And they're quite different systems, as we'll see. They do control different things. And part of our understanding of how they control different things derives from their projection pattern. Last time we talked about the upper motor neurons having a direct line to lower motor neurons, but we didn't mention the track that they take. And we did mention that they decussate, so at the I think it's the medulla level. So there's a crossing over of the upper motor neurons in the cortex, and they follow this tract that's called the corticospinal tract. And this is referred to as the lateral tract of the white matter of the spinal cord. And we'll see in an anatomical drawing of this in a minute. But basically they travel in a particular core of fibers down the spinal cord. The brainstem neurons travel in a different tract. So they're distinct systems. And this tract is called the anterior medial tract. Okay, so corticospinal tract takes the lateral tract to the lateral ventral horn. Okay? The upper motor neurons in the brainstem take the anterior medial tract and terminate in the medial ventral horn. Okay? And one of the other interesting things is that this travels down ipsilaterally, but then makes bilateral projections. So they're not strictly crossing over like the upper motor neurons in the cerebral cortex. Okay, so why is it important? So they both end up in this ventral horn of the spinal cord. So if we sort of drew, and there'll be a slide of this in a minute, we sort of draw a really terrible picture of the spinal cord. This is dorsal. Okay, and there's neuropill sort of in the middle, sort of butterfly looking. Okay, so here we have the dorsal part of the dorsal part of the horn, dorsal horn, and here we have the ventral horn. And we're further subdividing the ventral horn into a medial part. Remember, medial is toward the midline, and a lateral part, lateral toward the outside. Okay. So why does that matter? Okay. It matters because there's a mapping in the spinal cord of muscular control. And the mapping is this, that you have on the lateral side, you have control of the distal muscles, those sorts of skeletal muscles of your hand and your arm. And then on the medial side, you have proximal muscles. So these are sort of the near shoulders, torso kind of muscles. So you have this sort of distal to medial mapping, even at this level of the spinal cord. Okay. So you have these upper motor neurons going to that lateral area to control your distal appendages. And the brainstem areas are going to this medial area, which involves your torso. So how do you think that differentiates the sort of function of these two areas? Which one of these do you think controls, grasping? Obviously, the cerebral cortex. Which one do you think is involved in sort of standing and postural information? That would be the brainstem neurons going to the medial portion. Okay, so there's a real functional dichotomy here in what types of movement these descending projections are controlled. Okay. And it kind of makes intuitive sense, right? So we kind of think of the motor cortex as being involved in voluntary movement, throwing balls and so on. And we think of the brainstem as being sort of really fundamental, right? As breathing and cardiovascular function and all those things. And it's also important to this sort of fundamental thing of keeping you upright. Okay, so that's one way to kind of remember the difference between those. Okay, so here's another view. The anatomy really gives you clues into what the function of each of these divisions are. Here we have cortical inputs arriving via the lateral tract. So this is the lateral tract here and here. So you have these cortical inputs arriving. Remember, they've crossed over from the other side already and are arriving to the distal muscles. And the brainstem inputs are coming down and making these sort of bilateral inputs to the proximal muscles. Again, you can kind of remember this is bilateral because things along the midline, it's a little easier to kind of branch off and cross over. So, yes?
So is the brainstem always going to actually,
Never mind. Okay. I'm sure whatever you're thinking about, we'll probably come to. That's how it usually works. Okay, so what brainstem nuclei are we thinking about? There's a lot of them. Not all of them we're going to talk about. But the ones in particular we want to talk about today are the vestibular nuclei, which we've been introduced to before in our lecture on vestibular systems, and another one called the reticular formation. And we're going to learn just a little teeny tiny about the reticular formation. It's in fact a very complex and diverse collection of networks that have lots of different functions, and we're just going to kind of focus in on this particular motor function. And the idea is that these brainstem nuclei are involved in basically postural control. So that's kind of our theme for the brainstem networks today, is postural control. And two different kinds of postural control. We've already talked a little bit about this in terms of the vestibular function. So the vestibular nuclei provide feedback. You're starting to tip over. You're going to get input from your vestibular nucleus to tell you to straighten out. The reticular formation conveys a very different kind of information. It's actually passing on some feed-forward signals in terms of postural control. And we'll get into this further in a minute. But it's really this one system is providing feedback, and the other system is providing feed-forward control. Okay, and this is really considered kind of an indirect pathway, as opposed to the direct pathway of the motor cortex. This is an indirect pathway. It receives higher order signals and passes on signal to the spinal cord. All right, so let's just take a quick look at where these are. So here we have in yellow the lateral vestibular nucleus. Let me just remind you that this is a dorsal view of the brainstem and the diencephalon. You probably recall this from some of our anatomy lectures. Here's the diencephalon. Here are the colliculi, superior and inferior colliculi. And right here, it's not terribly obvious, but you see this sort of flat region right here. These are the cerebellar peduncles. Okay, these are these big superhighways that go up to the cerebellum that we've cut away so that we can actually look down the brainstem. So I took your head and removed your cerebellum, we'd be looking down on your brainstem right here. Okay, and this big hole here is the fourth ventricle. Okay, so the vestibular nuclei are located right here. I can tell you the auditory nuclei, the brainstem auditory nucleus is right next to it. You know it well. And then the reticular formation in this picture is this sort of red little column. Okay, and I'm not going to bother showing you sort of the whole anatomical shebang because it's not really that important for what we're doing, but it's a sort of extensive network of little networks, like a group of networks in this area. But it's here. Okay, it's in the brainstem. Okay, so these are the two nuclei, and the descending projections of these nuclei, these brainstem nuclei in general, are often referred to as bulbospinal. So from the bulb, the brainstem, this bulbous brainstem, to the spine. So whenever you see something like this, usually the first half of that is where it's from, and the second half is where it's going. Okay. So they form this bulbospinal tract to the lower motor neurons. The vestibular nuclei, there are two divisions. We don't really have to distinguish them here. But recall that these receive input from the vestibular nerve. And so this nucleus is both receiving sensory information and providing motor output. Okay, and then the tract here is the vestibulospinal tract. Again, where it's from, where it's going. The reticular formation, as we said, is complex and provides motor output, and it's called the reticulospinal tract. Very predictable. But in this case, boring is good. It gives you a clue as to what's going on. And then, you know, this is just to say there's lots of other things the reticular formation is doing that we're not going to discuss at all. All right. So what did I mean by? I think everybody kind of gets the idea of having vestibular feedback, postural feedback. It's the other half of the equation are anticipatory signals, right? So when you're walking, okay, and we talked already about how you're not really thinking about walking. But if you do, you have to lift your leg, right? One of the first things you have to do before you actually lift your leg is shift your body weight to the other leg, right? Otherwise, you lift your leg, you fall over. Okay, so you actually have to make movements before you make movements in order to maintain postural equilibrium. Okay, and so here's sort of a textbook example of how this happens. And so the task here is this person is trying to, pull on a handle that's attached. So they're going to, flex their biceps to pull on the handle. In order to maintain their positional stance, they actually have to make adjustments in other muscles, such as the gastrocnemius muscle, so your calf muscle. So in order to pull, you have to move your other muscles. Does anybody weightlift? Yeah? Yeah? Weightlift? So when you're using one of your muscles, you're not just using that muscle, right? You have to do a lot of other movements in order to compensate for the effect on your body, right? So you start pulling on one of those, what do you call it? Give me an example. Anybody have an example? Anyway, you know what I'm talking about. When you start pulling on one aspect of it, you have to do it. And so anybody who's, lifted weights in a weight room for the first time, where are you sore? Are you sore just in the bicep muscle? No, you're sore like all over because you've used muscles that you had no idea you were using. And it's because of this sort of postural control. So you have to do these sorts of counteracting muscles. Okay, so in this example, you have a signal. This is I think this is an EMG, yeah, gastrocnemius EMG. So you have a signal in your calf muscle before you even start pulling on your bicep muscle, activating the bicep muscle. Okay, and that's what we mean by this sort of feed-forward postural signaling. Okay, so the idea is that you have a central command that's going to initiate a limb movement, but at the same time, you have to do the postural adjustments. So you're sort of thinking ahead as to what you have to do in order to do it. Let me You're not really thinking, but your body's thinking. Okay, and then once you make a limb movement, this causes the postural instability, which then provides a feedback signal. So you have a feed forward and a feedback signal. So here's another way of thinking about this in terms of these pools of neurons. So here are our two brainstem nuclei, the vestibular nuclei and the reticular formation. You get an input from the cortex to the reticular formation that then provides an input to lower motor neuron pools. Okay, so this is your feed forward circuit. And then the vestibular nuclei get feedback. Sorry, this should be vestibular feedback. And that's added together into the lower motor, or activating maybe different lower motor neuron pools, depending on what you need to do. So we can think of this as these two circuits. This is the anticipatory postural control, which is a feed forward circuit, and a feedback circuit, the reactive postural control. Okay, and what I'm not showing here is that direct pathway. This is the indirect feed forward pathway. Okay, so just to kind of summarize what we're talking about, we have both indirect and direct pathways for motor neuron control. So there's two sets of the upper motor neuron pathways, and they have distinct roles. You have the motor cortex, which sends direct projections. It uses the corticospinal tract, descends in a distinct lateral tract, terminates in the lateral ventral horn, and controls these distal fine motor units. And then the brainstem nuclei really constitute an indirect pathway via the reticular formation that in turn projects to spinal circuits, descends in the anterior medial tract, terminates in the medial ventral horn bilaterally, and controls these proximal postural motor units. And these are driven by actually originate in different parts of the motor cortex. So this is this primary motor cortex system. The cortical inputs to the RF actually come from the premotor cortex. Okay, so the premotor cortex, let's see if I have this. Yes, so here we go. So here's your primary motor cortex. Oh, it's oriented. So this is the nose on this side. Okay. So remember, here's the primary somatosensory, then you have the primary motor cortex, and then just in front of the primary motor cortex, anterior to the primary motor cortex, is this premotor cortex. And the premotor cortex is sending signals to the motor cortex, but it's also sending these anticipatory signals to the brainstem. Okay, so in a sense, the premotor cortex is the boss of everybody. Okay. Okay, and so you can sort of dissect these two pathways apart and detect whether there's damage to these two different pathways, because if you, say, transect one component, this lateral tract, you can get very specific deficits. You can get deficits in fine distal moving, but people who can basically do postural movements just fine, can stand up just fine, but can't make physical movements. All right. Does anybody have any questions on that so far? All right, good. Okay, so now we're going to talk about the basal ganglia. And as we said, this is the primary ganglia that's involved in certain aspects of neurodegeneration that are very, very common. In particular, Parkinson's and Huntington's. So though as we see, Huntington's is actually kind of more widespread degenerative effects. And so this was kind of a nice aside. Okay, so here's the overview that we showed at the very beginning of the first lecture on motor systems. So again, we have lower motor neuron pools interacting with local circuits in the spinal cord, receiving sensory input, and then providing a muscle movement output. These receive descending input from these upper motor neuron units, the motor cortex and the brainstem centers that we just covered. Okay. And then in turn, there's another layer. Actually, there's two layers, two separate layers. And the one we're going to talk about now is this basal ganglia. And they have, somewhat different roles. I kind of hesitate a little bit because I think there's just a lot of unknowns here. The basal ganglia loops, as you'll see, are quite complicated. And I think we're really just sort of starting to understand their role in a lot of different cortical functions. We're going to talk about it in sort of this motor systems context, but they're involved in a lot of other aspects, probably much more than we can appreciate right now. And then next time we'll talk about cerebellum. Okay, so I want to kind of introduce, this is sort of the core concept of a basal ganglia loop. Okay. And the idea is that you have cortical outputs that go to the basal ganglia, and the basal ganglia in turn talks back to the cortex, but does it sort of via the thalamus. So there's kind of an intermediary middleman there. But this basic idea is that you have these loops. And the function of these loops is to basically gate the output of the cortex. Think about it. Okay, so you all remember this? You probably hated this in the first lecture, and here it is again. Okay, so this is sort of a see-through lateral view of the brain, and this is that basal ganglia, this sort of odd tadpole-shaped thing. So in the yellow is the basal ganglia, and what you can see in this sort of lateral view is the putamen and the caudate. So this whole yellow tail here is the caudate. It terminates in the amygdala, which we'll talk about in another lecture, and sort of nestled underneath the tadpole is the thalamus. So let's do a cutaway view. So we're going to cut through here in two positions. Okay, and look at a coronal section. Okay, but first of all, I just want to kind of get our terminology straight because it's a little hard to remember. That so basal ganglia refers to sort of three structures. It refers to the caudate, the putamen, and the globus pallidus, which we didn't see in that last slide, but we're going to look at next. Frequently, people refer to the striatum. Okay, the striatum is the caudate and putamen collectively. So those two together are striatum. Remember that. We're going to. These are just a few other players that we're going to talk about today that are not. They're not technically basal ganglia, but they interact with basal ganglia, and that's the subthalamic nuclei, the substantia nigra, and a particular component of the thalamus that we'll refer to as VA-VL complex. Okay, so this is another chunk of the thalamus. So the thalamus is a really busy place, right? It's got sensory inputs coming in, and then it also has motor outputs going out, and it has these basal ganglia highways going through it. Okay, and the thalamus is really, really important in this system. All right, so here's a cutaway through this more anterior section. Okay, you remember this. Come back to haunt you. Okay, but you get a second chance at it. All right, so again, this is the all the folds of the cortex, and the sort of tan here is gray matter, and then the whitish is white matter. Okay, you know all that. But sitting in the middle here, sort of the biggest chunk of this forebrain are these basal ganglia. Okay, and so we're going to show a bunch of slides, so don't worry if you don't kind of catch on just now. Here is caudate and putamen, so it's sort of this wedge shape right here, and it's bounded by a ventricle over here, and a white matter over here, and the anterior commissure down here. Okay, and then on the other side is the same thing. Okay. This white matter in between, that sort of cuts in between the caudate and the putamen, is called the internal capsule. It's really just a little highway of access. It's an important highway of access, but it has nothing to do with basal ganglia. Okay, but it's a good landmark to kind of tell the difference between these two. So you have this wedge of the putamen and a wedge of the caudate. Okay. I think that's all I want to say on this slide. if we look at the next slide, so now we've moved a little bit posterior, and now we've cut through a different part of this sort of 3D structure. So I think one of the most difficult things about neuroanatomy is trying to take these sort of two-dimensional cuts and recreate the 3D structure in your head. And it takes practice to do that. If it helps, in the back of your textbook, there are a lot of basically MRI series in a couple of different planes. So if it helps you, you can look at that, but you don't have to memorize that. But it might help you sort of visualize this. I'm going to show a video at the end of class today that'll also kind of help detect this. Okay, so in this section, okay, where is the caudate? So here, basal ganglia, and so here they refer to globus pallidus also as basal ganglia. That's the three together. Okay, so now the caudate is this little chunk right here. The putamen is this little archy chunk right here, and now we can see this little wedge of the globus pallidus. Okay, so it's changed shapes. So different sections obviously are going to have different shapes. But in this section, now we can see the thalamus. So here's the thalamus. It's sort of like little divisions in here. Okay. So I think next, so we talked a little bit about the internal capsule. And you can see this is internal capsule right here. So that has gotten really big. And that's because this is the superhighway that takes the axons from the cortical layers in and out of the forebrain. Okay. So I'm going to show you what this internal capsule is from another point of view. So these are all the fibers of the internal capsule. So this little flat bit here is the internal capsule that's cutting through the basal ganglia. And what we're looking at here is just imagine that you've sort of transected the brain, okay, and you're looking from the medial direction, and you've taken out the thalamus. And what you're looking at is this sort of fan-like projection of these fibers from the cortex. And I think there's ascending and descending components here, but they're all sort of in this fan shape. And I just think this is really cool picture. But that gives you an idea of where all those fibers are coming and going. But again, it has nothing to do with basal ganglia. It's just kind of a good landmark for our purposes. So here's the same cross section I just showed you, but a little helpful color. So again, here's putamen, here's the caudate on both sides in orange. Globus pallidus is this purple wedge. This is the component of the thalamus that we're interested in, the BA-BL complex. And then here you can make out these subthalamic nuclei. And we'll get to this in a few minutes. Okay. Another really important component in the circuit that we're going to discuss is the substantia nigra. Now the substantia nigra is not in the forebrain, it's actually in the midbrain. And I think there's some theory about whether this is actually basal ganglia that's been displaced for some weirdly evolutionary reason. But at any rate, here it is in the midbrain. And this is a cross section of the midbrain, and just as a landmark, well, not just a landmark, we're going to talk about this also, is the superior colliculus. So this is a midbrain nucleus involved in eye movements. And we'll get to that as we go along. Okay. So substantia nigra itself has two parts. One is called pars compacta, and one is called pars reticulata. Okay, and they're just colored two different colors here. So pars compacta is one layer, and pars reticulata is another. And they do different things. Okay, this is just a blow-up of that last slide, just to kind of point out a little better the globus pallidus. So the globus pallidus itself can be divided into two components, the external and the internal components. So internal meaning a little more medial than the other. Okay, so the principal inputs to the basal ganglia are cortical inputs that arrive in the striatum, the caudate and putamen. Okay, so you have this stream of inputs that arrive from all over the cortex to these two centers. They also receive input from the substantia nigra pars compacta. Okay, so a particular subdivision that happens to be dopaminergic. Okay, and I think all of you associate the sort of neurodegeneration of Parkinson's with the dopaminergic pathway. And we'll get into the specifics of that. Okay, the outputs are from the caudate and putamen to the next level, in particular the globus pallidus internal segment. So putamen right here goes to the next segment. Sorry. Okay. So it goes to the internal segment. It shows the external segment here. We're going to talk about that in a minute. It's not wrong, but we're going to ignore it for just a minute. Okay, so the ones that you want to think about right now is the caudate and putamen projecting to the globus pallidus internal segment, and then a second pathway is to the substantia nigra pars reticulata. So this is the different part. Remember, the compacta is the dopamine. This is the pars reticulata component. Okay, so we're actually going to kind of talk about two different pathways. They have very analogous function, but there are two of them here. Now, the basal ganglia cortical loop is the component that goes through the globus pallidus internal, back up to the thalamus, and back up to the cortex. Okay, so here's that loop that we talked about. Okay, so same thing, a little bit bigger. Okay, and just to kind of add another element, the cortex, we said, provides input to the caudate and putamen. Caudate and putamen send an output to the globus pallidus. This sends an output to the thalamus, which sends an output to frontal cortex. And for what we're going to term the motor loops, so the motor component of the system, this output is to motor or premotor loops. Okay, so there's a very specific loop to that. Okay, the principal cells in the caudate and the putamen are termed the medium spiny cells, okay, or MSN. Okay, and there are an enormous number of these cells, but 75% of them are these medium spiny cells. And here we're showing them projecting onto the globus pallidus, and they make a very, very strong connection. So this is the same sort of circuit. Here we have the cortical inputs projecting to the medium spiny neurons, and then you have the medium spiny neurons making a very strong projection onto either a globus pallidus or substantia nigra neuron. Okay, so I'm going to back up just for a second. So really key to this system is understanding which connections are excitatory and which connections are inhibitory, because this is fundamentally a disinhibitory circuit. Remember, we've seen disinhibition in a couple of different places. Retina, most notably, we talked about disinhibition using the horizontal cells and so on. Okay, and here we see it again. This is kind of a thing that we see over and over and over again. Okay, so there's a particular pattern here. So the inputs in the cortex are excitatory. So this little positive sign. Excitatory inputs to the cortex, from the cortex. The caudate and putamen have an inhibitory effect on the globus pallidus. The globus pallidus has an inhibitory effect on the thalamus. The thalamus has an excitatory effect on the frontal cortex. So if you can activate the thalamus, you can activate the cortex. And now we're going to talk about under what conditions that would happen. here's the circuit again, kind of a neural cartoon version. Okay. So here we have the striatum neurons. Remember, this is the caudate and putamen. We're just calling it striatum here. Projects to the globus pallidus, projects to the thalamus, projects to the cortex. Okay. The principal inputs to the striatum are these cortical inputs that we said were excitatory. Here's our inhibitory connection, excitatory. Okay. But the absolute key to how this system works is that the globus pallidus is tonically active. Okay? Those neurons are tonically active. Resting state, tonically active. Okay? If there's no activity in the striatum, these are tonically active. Okay? The fact that the global pallidus neurons are tonically active means that the thalamus that receives input, inhibitory input, are tonically suppressed. Right? So what happens if we shut off the globus pallidus? What happens to activity in the thalamus? It goes up. There's your disinhibition right there. Okay. So when is the striatum going to be active? Is it going to be more active or less active if it receives input from the cortex? More active. Okay. So the activity looks like this. At rest, okay, here we have striatum, globus pallidus, thalamus, cortex. Okay, at rest, the striatum is quiet, and the globus pallidus is active. That's suppressing the thalamus, which then is not exciting the cortex. Okay, so relatively speaking, it's not exciting its targets in the cortex. When the striatum is activated, it's getting inputs from the cortex. So the striatum cells are now active. They're inhibiting the globus pallidus. Okay? That removes inhibition from thalamus, which now fires, which then activates cortex. Okay, did I say that again? Activating the striatum, cortical activation of the striatum, inhibits the globus pallidus, which disinhibits the thalamus, which excites the cortex. Okay, so now this maybe makes a little more sense. This component is tonically active, and we're getting this series of activity. So activating the input signal from the cortex activates a particular part of the frontal cortex. Okay, you can kind of use the same logic in this pathway, right? So activating the caudate and putamen is going to inhibit the substantia nigra, pars reticulata. This was inhibiting the superior colliculus. So if you inhibit the substantia nigra pars reticulata, it disinhibits the superior colliculus, which is has motor outputs and can cause eye movements. Okay. So just to kind of summarize, the really key concept here is that there's a tonic activity of an inhibitory nucleus or set of neurons that are acting like a brake on motor activity. Okay, right? You're not doing all of your potential motor activity all the time. You have to basically suppress motor activity. Okay? And when you want to activate something, you have to disinhibit the brake. So you have to remove the brake, and that causes disinhibition and allows you to make motor activity. Okay, so the basal ganglia circuit is the brake. Okay, in the basal ganglia circuit, the brake is this globus pallidus for the cortical loop, or the substantia nigra pars reticulata for the superior colliculus. All right, so example. I'm going to show an actual example. And because the superior colliculus system it's a little easier to kind of read out what's going on. We're pretty good at measuring eye movements. The animal can be sort of sitting still while it's still making eye movements. So it's a very tractable system for understanding this. And this is why we're using this as an example rather than the cortical loop. Okay, so we're going to show how this system works in the superior colliculus system. Okay, so the task is this. The monkey has to sit and make a fixation. And when a little light comes on at some spatial position, it has to make an eye movement. Okay? We're tracking its eye movement. And when it makes the right eye movements, it gets a little bit of juice. So rewarding. So light's doing this. Okay. So here we have time. Here we have the target onset. So the little light turns on a particular spot. And here we're measuring the horizontal and the vertical eye position. So you've got to move a little, maybe a little to the left, a little down, make that eye movement. Okay. If you record from the superior colliculus at the same time, you get a very nice signal. So here's time, and when the animal has to make an eye movement, you get a firing right here. Okay, so the superior colliculus itself encodes the direction and location of those eye movements. And you can show this by stimulation experiments and by recording and so on. Okay, but we're kind of interested in this basal ganglia control. So again, here's our cross section. Here's our recording from in this case, the caudate. Okay, the caudate is sending a projection to the substantia nigra pars reticulata. This is the purple, and so we can record from the caudate and the substantia nigra and the superior colliculus all simultaneously. And what you can see is that at the right time you have activity, excitatory activity in the caudate. Here you see this tonic activity in the substantia nigra. So at rest it's just firing away. Okay. When it receives an inhibitory signal from the caudate, it shuts off. This relieves the inhibition in the superior colliculus, and you get firing. Okay. If you line up these two right here, you can see that these are timed quite nicely. Okay. So here's just a good example of that you can see how these disinhibition circuits are working. Okay, so this saccade experiment is just a nice example of this disinhibitory function of the basal ganglia, gating motor activity. Okay, and we can apply all the same principles to the frontal loop, except the ultimate trigger is in the frontal cortex. Okay. So now, we're going to add to this. Okay, so what we've been talking about is known in the field as the direct pathway. I know it sounds a little complicated. That is the direct pathway, and now we're going to talk about the indirect pathway. Okay, so wait, there's more. The indirect pathway are antagonistic. Okay. Just like we had antagonism of muscles, right? And antagonism of center surround circuits. Here we have antagonism of these basal ganglia nerves. Okay. And it makes sense, just sort of from a general intuitive perspective. Right, you don't want a general gating signal of do movement now. You have to do a particular movement while suppressing all of your other movements. The animal has to make a saccade to one position and suppress movements to all of the other positions. And that's what the indirect pathway is doing. Okay, so here's the direct pathway. Okay, here's that loop again. Cortex, striatum, composed of the caudate and putamen, inhibiting the globus pallidus, which is tonically active, inhibiting the thalamus, which then can't inhibit the frontal cortex. If you activate the cerebral cortex, you activate the caudate. That suppresses the globus pallidus, that disinhibits the thalamus, and excites the frontal cortex. Okay. And here is where we're going to start talking about the dopaminergic inputs. Okay, so the substantia nigra pars compacta we talked about provides a dopaminergic input. Okay. So now we're going to build on the direct pathway and talk about the indirect pathway. And you'll understand why we say indirect, because there's another step involved. Okay, so here's the direct pathway, right? Okay, you guys are all experts in the direct pathway now. So earlier we talked about the caudate putamen has outputs to the globus pallidus. So it has outputs to the globus pallidus internal segment, but also the external segment. And you saw that in some of the earlier drawings. There were two outputs to both segments. Okay. The external segment has an inhibitory connection, the external segment has an inhibitory connection to the internal segment. So here we have another sign reversal. Okay? So the inhibitory signal from the caudate putamen is flipped in sign if you go through one more layer. Okay? Let's see if this is right. Okay, let's say that one more time. So the caudate putamen has an inhibitory effect on the globus pallidus internal segment, but it has an inhibitory effect on an inhibitory input to the globus pallidus internal segment. So this means that this is an excitatory connection. Flip the sign twice. Okay? And that's sort of the fundamental antagonism. So outputs of the striatum is both inhibiting and exciting the globus pallidus internal segment. Okay. And then that's having whatever effect on the thalamus. All right. So just to add one more layer, the external segment has also an inhibitory effect on the subthalamic nucleus. Remember we said we were going to come back to this. This has an excitatory input to the internal segment. So now this again is actually the same type of signal. So negative, negative means positive. So this is negative, negative, positive means positive. Okay. So both of these signals has a positive effect on the internal segment via the indirect pathway, and a negative effect on the globus pallidus via the direct pathway. Yes?
Could you just explain why the system was designed like that?
Why? Why was the system designed like that? I can't explain that. And this is, I think, where we're still trying to learn how the system is working. What we're not really showing here is the precise mapping. You know, we're talking about each of these as these boxes, right?
Columns.
And there's, so there's basically mapping of cortical inputs map pretty precisely to the caudate putamen. These map pretty precisely onto the thalamus. The thalamus has very particular connections to the cortex. And this is kind of where our understanding breaks. Yes?
So do you think that maybe the globus pallidus, do you think it's targeting neurons that map directly to the caudate, or do you think it might be targeting like neighboring columns in a way to like sort of locally amplify or like clarify the signal?
Probably all of the above. So the question was, do you think that, basically, is there a complete loop here? Is that, if I rephrase it that way, is there a complete loop? So, you know, spot A in cortex projects to putamen, spot A in putamen projects to spot A in internal segment, and then you get feedback directly back to spot A. Maybe, maybe not. I think the key here is that there are competing circuits, okay? And you're trying to, you have this balance, right? So you have a direct pathway that wants to activate, and an indirect pathway that wants to suppress, and adjusting the balance between those two, I think, is the key. But we don't really understand. There's probably somebody who's got a very complex model for exactly how this is. You know, but say you want to have, say you have one circuit that's going to allow you to make an eye movement in one direction, and right next to that are the set of neurons that will make an eye movement on the other, and you have to take some sort of sensory information, where's the light coming from, and determine which position, which direction you want to move. And, you know, or maybe you want to assess how much reward you're going to get, or any other sort of behavioral factors. You know, how much motivation do I have? You know, if I make this eye movement, am I really going to get a reward, or is this kind of, you know, one of the tricky trials where they withhold? So there's, you know, an integration of information that has to happen in order to make those choices.
Okay.
And so you can think of the system, anytime you have sort of these antagonistic systems, you have sort of a fine balance, right? It's none of this is sort of, you know, labeled line, light, move, light, move. You have, do I want to move? Do I move in the right direction? You need feedback. Did I move in the right direction? And all of these are probably coming into play here. Yes?
So from the caudate and the putamen, is it the indirect and the direct, do they happen at the same time every time, or do you choose between either the indirect is happening or the direct is happening?
So the question is, are these direct and indirect pathways happening at the same time? And I can't answer that. I don't think it's really well understood. And I don't have an example to show exactly how that happens, but you could imagine sort of two parallel motor pathways that are getting different degrees of activation. And so whichever one, maybe one wins, is going to help suppress the others.
So the signal from the caudate and putamen to the internal and external, they're not the same degree on both.
I don't think it's well defined.
Okay.
You could certainly, if they were exactly the same degree, then you're basically going to get canceled out signals. If you have, say, one pathway, the external segment is going to suppress the internal segment of the other motor pathways, you could certainly imagine a system where that would happen. So basically, you know, we haven't really talked about it. You have like a winner-take-all system where, you know, the one pathway that's most excited then suppresses all of the other pathways, and that's probably a certain component of it. I didn't actually mention. So we didn't talk about in detail how these projections are set up. I sort of mentioned that there's a mapping there, right? And it has something to do with you know, where the neurons in the caudate project to a particular point in the globus pallidus. And there's a very strong projection here. There's a lot of convergence with this strong projection, but there are lots of collateral projections as well. So it's branching out and contacting neurons that are not in its sort of its direct labeled line. So there's a lot, you can see there's a lot of potential here for interactions. And there's probably a certain amount of sort of lateral suppression going on, you know, which makes intuitive sense, right? You want to select one motor action and suppress all the others. So you can think about it that way. And unfortunately, I probably don't have a very satisfactory example to demonstrate that, but that's probably what's going on. Yeah, another question.
Yeah, the diagram here is here as though since both the external and internal segments of the globus pallidus are tonically active, that when you get some transient input from the caudate, the external segment reduces its inhibitory effect on the internal.
Yes.
So do we know anything about the degree of the effect relative to the transient input from the putamen? Because it would have to be less, otherwise.
Yeah, these are great questions, by the way, because I can tell that you're following the pathway and thinking about the consequences. So I just want to say that's really great. Okay, and I'm sorry that I don't have really good answers for these great questions. But I think those are really great questions. So you can imagine, right? So if you get an output from here, okay, and this is inhibiting the external segment, which was tonically inhibiting the internal segment. So we said earlier that the really key function is that this internal segment is tonically active. Okay. In order for that to be tonically active, this has to be only partially suppressive. So if I would say that both of these, think of these both as being tonically active, but the external segment is not sufficient to prevent activity in the internal segment. So let's just sort of stipulate that. The internal segment is tonically active. It's receiving inhibitory input, but it's not tonic inhibitory input, but it's not sufficient to suppress it. This is just like the horizontal cells in the retina, right? So those horizontal cells were in dark, tonically active, but not enough to completely suppress. But if you remove that inhibition, you get excitation. So that's a really key point. All right. So, okay, so we have direct pathway, direct disinhibitory pathway, indirect pathway that flips the sign and is now relatively provides an excitatory signal to the internal segment. So this pathway provides ultimately an excitatory signal when the cerebral cortex is activated. So, sorry, let me walk through that again. Activate the cerebral cortex, activates the caudate putamen, inhibits the external segment, removing the tonic inhibition and exciting the internal segment. So activity in the cortex in this pathway suppresses the internal segment. In this pathway, excites the internal segment. And now if those were equal, you would have a cancellation of whatever the input is. So probably there's subtleties in terms of, which if you shift the balance, you're going to shift toward suppression of the internal segment, which activates the motor movement, or excitation of the internal segment, which suppresses the motor movement. Yes?
I was wondering if you could clarify, does the D1 affect the disinhibitory pathway?
So we're going to talk about that next.
Okay.
Okay, because that's where the dopamine comes in.
Okay.
All right. Okay, so I told you that substantia nigra pars compacta is dopaminergic and provides a dopaminergic input. But it actually provides, the same dopamine has different effects depending on the receptor. So there are two receptors, D1 and D2. The D1 provides an enhancement signal for the direct pathway. So this little arrow here is meant to say enhancement of the direct pathway. This little arrow and negative sign is meant to say suppression of the indirect pathway. And we'll say this all over again. I think in the next slide. Okay, let me just skip to this slide for a second. So this dopaminergic projection to the striatum acts through these two receptors. So remember how we talked about how the effect of a neurotransmitter depends on the receptor, right? So you can have transmitters that could be either inhibitory or excitatory, depending on what the receiving end says about it. Okay, so this is a good example of that principle. If a neuron has a D1 receptor, it's enhanced, and the D1 receptors are associated with the direct pathway. Okay, if it has a D2 receptor, which is inhibitory, this is a characteristic of the indirect pathway. Okay. So dumping dopamine on is going to have a different effect depending on the receptor that's being expressed. Okay. And then just for completeness, the reason it has a different effect is that the D1 receptor activates a second messenger system that increases cyclic AMP, and the D2 receptor has a second messenger system that decreases cyclic AMP. Right? So the reason those receptors have a different effect is because their second messenger pathways do different things. All right. So then this dopaminergic signal is going to basically shift the balance between that direct and indirect pathway. Right? So we've set this thing up to have this balancing act depending on the cerebral cortex, but adding dopamine can enhance the direct effect and suppress the indirect. So this is just kind of a text explanation of the direct pathway and the indirect pathway. So the idea is that this direct pathway triggers the motor plan you want to do. Okay, saccade here. The indirect pathway suppresses any of the unwanted motor plans. Don't saccade over there, or here, or here, or here, or here, or here, right? Almost anything else. You want to do that one thing. Okay. And in that sense, the two paths are antagonistic. Okay. As we said, it's not really that you're getting a negative signal and a positive signal and everything's kind of canceling out, and that doesn't make any sense. What's happening is that you have this antagonism where you have to make a selection. Okay. And different factors can bias what selection you make. You have that cortical input that's going to have an effect, you know, which part of the caudate pancreas is being activated. And then you have this dopaminergic signal that can also bias. Okay, so just to this kind of walks through the same thing in words. The direct pathway ultimately inhibits the globus pallidus internal segment, suppressing GP activity, disinhibiting the thalamus, which is then excited and activates the motor cortex triggering action. The indirect pathway disinhibits the GP internal segment via the GP external segment. Okay. And then secondarily, it excites the GP through disinhibition of the subthalamic. So kind of walk yourself through that. And how are we doing on time? All right, so we're going to stop right there, and we'll talk about Parkinson's and Huntington's.